Thank you very much. Just want to, yeah, I can hear the microphone's working, so don't have to ask.
Yeah, I think you should be okay. Can you just point to this?
Meghna, the screen is not full screen.
Yeah, okay. Let me check. Can you please get the pointer?
Yeah, I can.
Let's do that. I think let's go with. A minute to get the technology working. And now it should be full screen.
Yes, it is.
Yeah, good. I think I decided to do the screen. Can you use the top down, up down bar till the pointer comes? Yeah, yeah. Or you can use any of these buttons. Is that okay? I'll just get you the pointer in just a few minutes.
That's fine. That's fine. Well, thank you. Thank you for the kind introduction. Thanks all of you for coming here or online to hear a little about what we're doing. And the focus of what I'd like to speak about is shown here in the title. So we're interested in translation of biomedical microtechnologies from the lab to the clinic. You'll see the microtechnologies that we're working with. Really, what the emphasis is really on is how we go about thinking about starting out with some idea and doing work in the lab and trying to get it into the clinic. The clinic being either clinical trial or even with approval for use in clinical medicine. As mentioned, I'm involved with starting companies based on the work we've done. The university makes me formally disclose to you that I could make money off of the technology, so I'm hopeful for that. But today we're talking more about the science, and let me just present a general challenge, which is if we want to administer a drug or a vaccine to the skin, there's a barrier in the way, and that's the stratum corneum, the outer 10 or 20 microns of the skin. So what do we do about that normally? We take a hypodermic needle that measures millimeters to centimeters, and we stick it in, and we make an injection much deeper than the barrier. So the thought is, why do we have to do that? Why not make needles very small and make them long enough to cross the stratum corneum barrier and get into the body, but they don't necessarily need to be longer. Sometimes there is a reason to make them longer, but often there isn't a need. And that allows you to do things like have a patch that you just stick on the skin. It doesn't require training, it doesn't hurt. And that very simple concept of taking a big needle and making it small now enables us to perhaps do some new things. And so that is really a guiding concept in what I'm going to be talking about for the rest of the time. So the microneedles could take a few different forms, and if you were at my talk earlier today, then a few of these slides are replicated, but quickly they will all be new. So there are microneedles that could be used in various ways. One of them is as just a piercing structure. So you take these small needles, you stick them into the skin, you pull them out. There's now some holes in the skin. Holes are of micron dimensions. Molecules are of nano dimensions. So the molecules can go readily through these holes, and now you get the drug to enter the body more effectively. Another approach could be the coated microneedle. So now you take your drug formulation, you put it onto the surface of the microneedle, you stick it in, and that coating comes off quickly. So unlike in the first scenario, it's a slow absorption over time through the holes. In the second coated needle scenario, it's a quick delivery, patch in, a few minutes, patch out, and all the drugs can deliver. You could make the whole microneedle out of water-soluble material. So now you press the patch on, and the whole needle dissolves and disappears, so there's no sharps waste when you're finished. And then finally, the microneedle could be more like a hypodermic needle. It could be a hollow needle, liquid formulation now. You flow that liquid formulation into the skin through the bore of your hollow needle. So these have various strengths and weaknesses, and I'd like to...
You can use it now with the pointers. Yeah.
I'd like to give 5 short stories that give some examples of these different microneedle types and what we might do with them. So the first one that I want to start with is the dissolving microneedle patch. So that's using these kinds of water-soluble microneedles loaded up with a vaccine that we can press to the skin. And instead of having doctors and nurses give shots, we could have lesser trained personnel administering patches to the skin. We could use the solid microneedle approach, and here I'll talk about what we call star particles. The motivation here is that if we want to treat large areas of skin, making big patches and lots of patches is complicated, and so we can put the microneedles onto particles and rub them on the skin, and that could enable delivery over large areas, over variable areas, and the experience would be much like just what you normally rub on your skin. We have a piezoelectric microneedle electroporator. So I'll say a little more about electroporation for those who aren't familiar, and explain why if we turn these microneedles not into a drug delivery system, but into a microelectrode, that could enable us to do electroporation in the skin, and that could have advantages such as getting rid of lipid nanoparticles for mRNA vaccination. Another variation on this solid microneedle is to use the microneedles in reverse. Rather than making pathways into the body to deliver a drug, we can make pathways out of the body to collect fluid out of the skin. And I'll explain why I think that's interesting. And then finally, we'll go to the hollow microneedle and make a big switch. We're not going to be in the skin, but we'll move to the eye. And I'll talk about how we could enable suprachoroidal injections using this approach and why that could be of interest. So that's the roadmap. And the first story will be a little bit longer than the others. I think we'll get through in time. Just small advertisements, if you will, on each of these topics. So let's start with this first one on the skin vaccination by microneedle patch. So here's a picture of some microneedle patches in the foreground and needles and syringes in the back. We can take a closer look at one of these microneedle patches, and here it's made bigger now for you to see. You can see here are 100 microneedles, each with some green dye in them to help you visualize. There's a shiny adhesive around the periphery to stick to the skin. There's a tab to hold on to so that you can handle it easily. And then finally, if we take a still closer look, so these are the solid dissolvable microneedles. You can see the individual needles. They're measuring hundreds of microns in length. So get into the skin, but not go terribly deep and avoid pain that way. We've loaded the green dye, which represents the vaccine, selectively into the needles, not in the backing. That's the part that gets delivered, so that gives us better efficiency of delivery. So what do we think about in terms of this development? Well, one is patient administration. Minimally trained personnel would be able to administer these patches. So you can imagine if a vaccination campaign needs to be done, if vaccination needs to be done in remote locations where there may not be a clinic that's there, we could now have people with a little bit of training be told how to use these patches, and now you would be able to vaccinate in settings where otherwise it would be much harder. No applicator involved, just your thumb, no reconstitution. That is the dried vaccine has water added to it before injection. That's the reconstitution process. We have a dried vaccine, which gives it greater stability, but we don't have to add water to reconstitute because it reconstitutes in your skin, delivered in a dry form. Painless delivery, no fear of needles, and so that not only makes kids not cry, but just makes people in general more receptive and accepting of the technology. The manufacturing process looks something like this. So we start out with a master structure in the shape of the microneedles, and then we make a mold off of that master structure. The mold is usually out of a silicone, PDMS. We can make the master structures. These days, we typically 3D print them, although other technologies could be used as well. So once you have this mold, then you cast a solution onto the mold that fills the mold cavities, a solution that would have in it your antigen, your vaccine, as well as other materials to stabilize that vaccine during the drying process, it would be dry, and also to give the microneedle mechanical strength. So that usually means that there's some polymer in there, and there's probably some sugar in there. The formulation varies based on exactly what it is you're encapsulating and delivering to maintain its stability. We then do a second cast to create the backing layer. So this doesn't have the antigen in it. That's how the two-cast process gives us the vaccine only in the microneedles and not in the backing layer. And then once everything is dried, we can remove it from the patch. So at one level, it's a very simple process. You mix up two solutions, you cast them both and let them dry, and you're done. There are a lot of details, of course, to do this well, and especially to scale it up and then do it well in the millions of patches. But it is a process that we think does lend itself to being done in a conventional kind of pharmaceutical manufacturing setting. And as I'll be mentioning in a little bit, that setting may just be down the road at the Serum Institute. We did a clinical trial that I'll tell you about using their vaccine. So for transportation and storage, it's a small package size. Basically, we get rid of all the water, which is most of what a vaccine is, and just keep the antigen and a little bit of excipient. So needles and syringes and vials and biohazardous sharps waste and cold chain storage, all those things can go away if we've designed our patches right. Waste disposal, there's no sharp waste. If the microneedles dissolve, they disappear. There's nothing sharp that remains. It's not possible to reuse it, and whatever disposal volume is left is reduced compared to conventional injectables. So these are some of the motivations. Let's jump into results from 2 clinical trials that I'd like to share. A bunch of work led up to this that I'm glossing over, but let me go to the end product. You'll see in the other vignettes I'm going to tell, they start at an earlier stage, mostly because they haven't gotten to as late as. So in our first clinical trial, this was a phase 1 clinical trial on influenza vaccination. It was in 100 healthy adults from the Atlanta area. We used an activated influenza vaccine by Seqirus. Seqirus used to be the Novartis influenza vaccine, and they're one of the major flu vaccine manufacturers. And we had 4 groups of equal size. We had our positive control, intramuscular vaccination by nurse. We had our 2 study groups, the microneedle patch vaccination, either by nurse or self-administered by the subjects. They saw just a few PowerPoint slides, and that's the extent of their training, and then they carried out the vaccination themselves. And then there was the placebo patch negative control administered by nurse. The study was a patch developed at Georgia Tech, study at Emory University Medical School, which is in Atlanta, and NIH supported it. So I'll just jump to the key results. This is showing the seroconversion. So this is the immunogenicity of the vaccine. And let me guide you through this a little bit. You can see there are 3 clusters of bars here. So there's the H1N1, the H3N2, and the B strain. So as you may know, today there are 4 strains of flu vaccine. There are 2 B strains. At this time, trivalent, not quadrivalent vaccine was the norm. So that's why you see the 3 different antigens that we're assessing. And then the different bars are, first one is the intramuscular vaccination, the next 2 are the 2 microneedle patch groups, and the last bar is the placebo. The y-axis shows you seroconversion, so that's the percentage of the subjects whose antibody titer went up by a factor of 4. It's a little more to that definition, but basically that's what it is. And so that is considered a successful vaccination if seroconversion occurs. So what you can see is for the 2 A strains in the 3 vaccinated groups, we're getting good seroconversion, 80% or more, and it's statistically indistinguishable between the 3 different vaccination groups. Obviously, the placebo is lower. So that shows that we can incorporate a vaccine into a microneedle patch, we can administer it to human subjects, and we get the desired response. When we look at the B strain, it gets a little more interesting, because you can see that for the intramuscular vaccination, the seroconversion was relatively low. And that wasn't a mistake of the trial, that was expected. The B strain is often less immunogenic than the A strains are. But what is really interesting to note is that by microneedle patch, we're getting as much as double the seroconversion rate. So there's another piece to the story that I didn't mention. We are, of course, vaccinating in the skin, not in the muscle, and that makes a difference immunologically. There have been a variety of studies that have shown in humans and even more in animals that if you put a vaccine into the skin, oftentimes it's more immunogenic than if you put it into the muscle. And that probably has to do with the fact that the skin is our interface with the outside world. The skin is ready to discover pathogens and mount immune responses against them in a way that the muscle is not. It's very artificial that a pathogen suddenly appears in the muscle. That's a feature only of the hypodermic needle. So as a result, there are Langerhans cells in the epidermis, those dermal dendritic cells. The dermis is a lot of lymphatic drainage out of the skin as well, and these things all come together to make the skin a more immunologically active site for vaccination, and we think that is the explanation for the improvement here. From a safety perspective, we looked at systemic safety signals, and they were few, and they were the ones you might expect. Some people have a cold, they feel like they have a cold, or that kind of a thing after they get a vaccine, and there was no difference between those among the various vaccinated groups. So really where the interesting part was locally at the site of vaccination. That's where we could see some difference, and often we call that reactogenicity. So what is seen here is on day 0, you see a close-up view of the wrist. We applied these vaccine patches to the wrist, and perhaps you can make out there's a little square in the center there. That's where the needles have just entered into the skin, and around it there's just a mild pinkish erythema, and that's from the adhesive that's just been ripped off the skin. Really has nothing to do with the microneedles per se. You come back after 2 or 3 days, and there's a faint evidence there. You can make out just a little mild erythema at that site. A little over a week later, subjects came back, and finally day 28, and there was nothing to see on the day 28 visit. I'm not showing the pictures, but if you look at the equivalent pictures for the placebo patch, day 0 looks the same. Day 2 to 3, you see nothing. And afterwards, you see nothing with the placebo patch. In fact, I know from other studies who've done placebo patches, even on day 1, you see nothing. And so the microneedle puncturing into the skin will cause a very mild transient erythema for hours, maybe a day, and goes away. What you're seeing here at these later times is literally the immune response. Vaccine has been put in there, and the immune system's coming in and doing something about it. So from a scientist standpoint, this is great. We want to see a little erythema in the skin because it means the vaccine's working. In terms of the characteristics that were notable, tenderness was reported in the microneedle patch vaccination group and the intramuscular vaccination group at about the same rate. So we didn't see a difference there. Erythema, skin redness, or pruritus, itchiness, both of those were greater in the microneedle patch vaccine groups compared to the intramuscular. Probably there was erythema in the muscle in response as part of the immune response. You just can't see it. You can see it in the skin. Pruritus, there are no itch receptors in the muscle. There are itch receptors in the skin, so that can explain that difference. And finally, the pain was greater in the intramuscular vaccination than the microneedle patch, that being the pain at the time of vaccination, as well as that sore arm you can sometimes get afterwards. That level of pain was also higher in the intramuscular group. Overall, there were no real safety concerns that came out of it, just some of these minor kinds of effects that may or may not affect acceptability by patients, but not a safety concern. So that led us to doing a follow-on study where we looked at measles and rubella vaccination, and that was just published earlier this year. And our motivation comes from this. So let's first look at measles. 25 years ago, there were 1 million kids dying every year from measles. Then there was intensive vaccination that was done. Money went into that, vaccination campaigns were done, and there were remarkable effects. That was lowered by an order of magnitude over a roughly 20-year period. Right now, perhaps in part due to complications with healthcare infrastructure during the COVID period, it's actually come up a little bit. It's a little over 100,000 now. But it has stabilized at about 100,000 now for many years. So what that tells us is intensive vaccination can make a big difference because the measles vaccine actually works incredibly well. You give a single shot of it, and most people are protected for life from a single shot. They give 2 just to get everybody. But you can only go so far. You pour all this money in, you get all these vaccines out, but then you hit the wall at 100,000 deaths. So 100,000 kids dying every year. And it's for lack of access to this vaccine. So we think that there could be a real advantage by making the vaccine more easily accessible by, for example, getting it at least in part out of the cold chain, by enabling minimally trained personnel to administer the vaccine. And the rubella story is a quite similar one. The intensive measles vaccination was generally accompanied by intensive rubella vaccination at the same time, and an order of magnitude drop in rubella cases as well. So with that in mind, we developed a microneedle patch for measles and rubella vaccine. And as I mentioned, the vaccine, well, started here, got shipped across the ocean to Atlanta where we developed it. So it's Serum Institute's vaccine. And we carried out a phase 1/2 clinical trial and that was carried out in the Gambia in West Africa. And the involved parties were the MRC, Medical Research Council, MRC Council, pretty sure.
Medical Research Council.
Council, yes. Medical Research Council, which is a UK organization. They have had a clinical trial research outpost in the Gambia for some decades. So that's where the study was done. Micron Biomedical is the company that is commercializing this technology out of Georgia Tech. So they made the patches. Centers for Disease Control and Prevention helped in the study design and did all of the serology. Gates Foundation supported it, and Serum provided that.
When was this roughly?
The study was done in 2023 and published in 2024.
Okay.
Basic design is it was a phase 1/2 study, so we're looking at safety as well as the immunogenicity in the target population. It was randomized, active control with a double-blind, double dummy design. So what does that mean? It doesn't mean we're done. It means that every subject in the study received a patch and an injection. We couldn't blind the study if we didn't do that, because everyone would then know who got a patch and who got a shot. So everyone got both. Half of the people got a shot with vaccine and a placebo patch. Half got a patch with vaccine and a placebo shot. So we could keep it all blinded for everybody involved. We followed an age de-escalation design. We started out with healthy adults just for safety before we went into kids. When that checked out, we then went to the toddlers, the 15 to 18-month-olds. These were kids who had gotten their first measles and rubella vaccination just through the normal vaccination process in country, and we gave them the second shot or the second vaccine. Only half of them got the shot. And then if safety was okay there, we moved to the infants who got then their first vaccination. We looked for local and systemic adverse events. So there's blood laboratories done after 1 week. We also followed unsolicited adverse events out to the study durations. There's a 6-month, half-year study duration. And we looked at neutralizing antibody titers at 2 time points. Okay, so what did we find? So in this case, the last time was seroconversion. In the case of measles and rubella, what is used is a different term, which is seroprotection. The seroprotection refers to the antibody titer that is known to be protective. So that's an even better thing than seroconversion. Seroprotection, there's so much data on measles and rubella vaccine that we actually know what titers needed to be protective. And you can see the baseline, there was some just background at the 2 different time points where we measured the red microneedle patch and the blue subcutaneous injection. You can see that they're essentially the same. We got nice responses and no difference based on the route of administration. Story is similar or even better with the rubella, 100% seroprotection in all vaccinated people, which was everybody in the study. So from an immunogenicity standpoint, looks good. From a safety standpoint, again, the local reactogenicity was the place to look. From the systemic safety, actually there were a lot more adverse events reported because these were infants and toddlers in West Africa, not healthy adults in Atlanta. So and people got all kinds of various illnesses that kids get, but none of them were greater in the microneedle patch group compared to the subcutaneous. So that was all a wash, and most of those were deemed to be unrelated to the vaccine by the study organizers. So again, reactogenicity is where we're looking right at the skin, and these are the results in the infants, the most sensitive group. Similar results in the others. We looked for tenderness, and so the upper 2 graphs here are for the microneedle patch, either when the patch had the vaccine or didn't have the vaccine, and then correspondingly we have the subcutaneous injection site if the injection was with or without the vaccine. And well, there's nothing to see because tenderness was not reported essentially by anybody. We then look for erythema, and now we do see something. There is some erythema seen in a small fraction of the subjects, and it can last out for a number of days. We see it here actually in the placebo group extending out over time, as well as in the vaccination group. The erythema incidence here is lower than we saw in the flu vaccine study. It's a different vaccine, different patch formulation, different study population. So there are various differences. I think an actually critical difference is that the population in the Atlanta study was mostly white, The population in this study was 100% Black African, and so it's just harder to see erythema in darker skin. And the thing that was most notable was induration or a swelling at the site. And here you can see a distinct difference where the vaccine patch group looks different from the other ones. And notice that the induration kicks in late, after more than a week. I'm not showing the data, but if you look at the same thing for the toddlers who had been vaccinated before and were getting a second dose, you also see the similar induration, but you see it immediately. So the toddlers were primed and their immune response kicked in immediately. These are not primed, so the immune response is delayed. It has to build up over time, and then it kicks in. So once again, we believe strongly that what you're seeing here literally is the immune response to the vaccine. So not of a safety concern, of a possible just cosmetic or other kind of concern to somebody who receives it. So that's the end of the first story, and I warn you, that's a little bit longer one. The stories will get more concise as we move along. And what's motivating us here now is not, actually, let me say just one other thing about the vaccination work. So the trial went well. Gates Foundation and others got very excited about it, so we're moving full force ahead. A manufacturing facility is being set up to do the manufacturing for what will be in the phase 3 clinical trial and ultimately, at least for initial commercial production, assuming we get there. We're planning the phase 2B clinical trial will be in the first half of next year. And so things are marching along. Now, sometimes we'd like to do, yes, please.
Can you go back to previous?
Or maybe we can just ask the questions pointed right now so that we can, please go ahead, clarifications, but after that.
Did the material you use for microneedle also contributed to the introduction? Okay.
So the question for those online is if the materials, not the vaccine, led to the induration outcome. So I think the answer is no, and the reason is twofold. One of it is just the data itself, right? You look in the placebo patch site, we don't see the induration, a little bit, but it's where we really see that induration happening is when you have the combination of a vaccine and the microneedle patch together. And the fact that it's delayed in the infants and immediate in the toddlers, and that suggests it's an immune response to the vaccine. So in aggregate, that would be my interpretation.
Let's continue.
So moving to the next topic, which is an interest not in vaccination, but now an interest in dermatology. So there's a common issue, which is we want to get some kind of a medicine into the body and across that stratum corneum. It's not a vaccine to generate a systemic response, but it is some drug to generate a local response at some site of skin disease. What's special about skin disease is that it could be over a large area, could be over a variable area, and that's unlike vaccination, where we just want to get the vaccine in. It doesn't matter so much exactly where, it just needs to get into the body. Here, the spatial aspects are very important. So rather than making patches that are really big or cut to customized sizes or lots of patches, it just gets complicated to use patches in that format. We thought, what if we take the microneedles and put them onto particles instead of having them on patches? We can make lots of these little particles, incorporate them into a topical formulation, and rather than pressing a patch in, the rubbing action, just as you normally do, that rubbing causes these particles to roll and slide around and make little punctures in the skin, so that now a topical formulation with the drug in it can be absorbed into the body much more effectively. So here are some of those microneedles that we made. These are made out of ceramic material. They're made out of titanium dioxide. Titanium dioxide, it's a whitening agent in many creams that people rub on their face every day, and in cosmetics. It's in white paint on the walls. It's used all over the place, just not in star particle shape. They're more of a spherical-ish shape. So the way that we make this is we have a film of very small particles, so much smaller than this, of titanium dioxide that are held together with a binder. We take a laser and cut out the shapes. We then put that into a furnace and sinter together all of the little titanium dioxide particles. They fuse together into making this star-shaped particle. And the furnace, it's really nothing different than a pottery kiln. In fact, the first ones we made, we went to the student center on campus to the pottery place, and we used their kiln. We got our own kiln in the meantime. But so the process is a simple one to do. The main cost is you got to buy some lasers and then have them cut out your particles. So okay, we have these particles. You can see there are Mercedes-Benz brand with the three-arm star particles. And you can then incorporate them into a cream. If you just look at them, they look just like a little white powder. And then we can rub them on the skin. So this is with pig skin ex vivo, where we have rubbed the skin and applied a dye, sulfurotamine, onto the skin, and you're seeing histological sections, cross sections in the skin afterwards. So in the first column, there we just rubbed some gel on the skin that had no star particles in it. That's our placebo. And then we put sulfurotamine on, and you can see even after 24 hours, there's essentially no penetration of that agent into the skin. In contrast, if we take that gel and put star particles in it and rub that on the skin, and then look at the sulfurotamine penetration, you can see that there's extensive penetration deep into the skin. We did the same experiment with a bigger molecule, 4,000 molecular weight, and again, this large Dex strand was able to penetrate into the skin nicely.
Just a quick one on why is the star shape so important in terms of the penetration?
So we use so the question is why star particle shape? So we definitely want to have a microneedle. We need something to puncture.
Okay.
But there are two things. One is we want them to come in, the other is we want to come out again. So there's been some work where people have in the past, they've taken little sort of glass fibers that are kind of like two-armed star particles, but they have a mechanism to go all the way in. So if you look at the star particle geometry, inherently it has a kind of a stopper. When one of these needles goes in, the other two needles get in the way and don't allow it to go all the way in. So the needle goes in and then comes back out again. The elasticity of the skin, in fact, has it come back out. It doesn't stay embedded. All right, so we're able to get some fluorescent compounds in, and even, I mean, relatively large ones as far as skin permeability is concerned. We then look at some real drugs that might be of interest. So we're showing the amount of drug delivered into the skin. Notice a log scale on the y-axis, and we've got 3 different drugs, and we assessed it at 3 different time points. So the first one is 5-fluorouracil. This is a drug that is given topically on the skin to treat some skin cancer. And we were able to show that if we rub the star particles on the skin in the gray, as compared to the white where we didn't rub the star particles on, we're getting order of magnitude increases in terms of the amount of this drug that goes in the skin. We looked at methotrexate that's used to treat psoriasis. Typically, it's given systemically. There's significant side effects associated with that. So if we could deliver it locally to the skin, then we can get rid of those side effects. Bleomycin is sometimes injected into the skin to treat warts. So we could get rid of that injection. That requires some expertise and have some pain associated with it, and use star particles to deliver the bleomycin. We took a next step on this and did a little more work on the 5-fluorouracil now in the mouse model. This is a mouse melanoma model where you can see if we rub just gel on the skin, or we rub gel with star particles on the skin, but no drug. We rub drug onto the skin, but no star particles, or we use the combination, star particles plus the drug. In the first 3 cases, large tumors grew. In the 4th case, it didn't. And I'll show you the quantitative data here. The tumor volume in the first 3 cases always increased. In the case of the drug plus star particles, we were able to prevent tumor growth from happening. You may also know that there are fewer data points on some of these, and that's because the animal tumor had gotten so big that we had to euthanize the animal. So a very clear difference where we were able to enhance the delivery of the 5-fluorouracil with the star particles. The last part of the story is in human subjects. There's no drug here. This is just looking at tolerability. So we took those star particles, rubbed them on the skin, and then looked at the skin afterwards. So here's what it looks like immediately afterwards. Presumably there's not much to see. And it came back a day later, and again, there was nothing to see. We've done a follow-on study in the meantime where we rubbed star particles on the subject 10 days in a row, every day at exactly the same site, and there was no change of effect. Day 1 looked just like day 10. Subjects reported the sensation on day 1 was just like on day 10. So it looks like whatever we do to the skin, it reverses and repairs itself immediately so that every day you could keep applying it. So let's go to story 3, but a question first.
Yeah, sorry, because the previous one which you mentioned on the star, because the drugs vary in different molecular weights. So do you see any difference in the effect because you have a extra based as well as small or even a small drug? So do you see any changes or defects pronounced when you have a different molecular weight? Because the diffusion.
Right, sure. So the pore that we have created is micron scale. So as far as the pore being big enough to let a molecule through, it's big enough.
Okay.
Pick any drug you like, it's smaller than a micron. So the hole itself is plenty big. But the problem is you need to diffuse through the hole. So unlike a microneedle loaded up with the drug, you stick it in the skin. Here, you just made a hole, and the molecule still has to fuse and through the hole into the skin. And so that's where molecular weight matters, because diffusivity goes down as molecular weight goes up. So our delivery rate does decrease when the molecular weight is higher. But it's not because the pores are too small, it's because diffusion is slower. So let's shift to, again, something different, which is now the microneedle is serving as a microelectrode. So we're delivering electricity, electrons into the skin. We're not delivering drug into the skin, but we think that can nonetheless be useful for DNA and mRNA vaccination. So we think about those topics and we think about mRNA, everyone immediately thinks of lipid nanoparticles, and indeed mRNA administered that way has been remarkable and is a savior, and we can all appreciate it. But at the same time, it has some real limitations. The lipids themselves are expensive. The microfluidic process of making the nanoparticles is expensive as well. It's complicated. The frozen storage is a complication as well. Lipid nanoparticles is good, but they're not perfect. But that's what we have for mRNA. If you want to deliver DNA, then viral vectors is what's usually used. And again, they work well, but they have their issues in terms of manufacturing, in terms of safety, in terms of repeated use and developing immunogenicity to it. So viral vectors also have their limitations. There is another option that's widely used in the lab, which is electroporation. And I don't know, there may be an electroporator in one of the buildings around here. So you give a short electric pulse, which destabilizes the cell membrane, and then your nucleic acid is able to go into the cell. In fact, if you look at the literature from the early days of genetic engineering in the 1980s, electroporation was really the go-to method, because that's what they had. In the meantime, we do have other options, but people widely still use electroporation in the laboratory. So if it works well in the lab, why don't we use it in humans? Well, there's some problems. So it has been used in humans, but the problems are primarily twofold. One of them is it's a big, expensive, complicated device. If you want to buy one of these electroporators for your lab, you might spend $10,000 to buy one of them. And if you likewise want a device in the clinic, you can see here, it's on a stand and this big thing. So it's expensive, it's big, it's complicated to use, you need expertise. And so that's one barrier. The other barrier is, well, I visited a clinical site where they were involved in a study of COVID vaccination using a DNA vaccine with a commercial electroporator, that I think maybe this one. In any case, they said, yeah, their standard procedure was that they always needed 2 people. The first person holds down the arm, the second person gives the electric pulse, and the first person holds down the arm so that when the electric pulse excites the nerves in the arm, the violent twitch that results doesn't cause the arm to go moving through the air. So it hurts, it stimulates nerves, and so you can do that in the lab, but when you start doing it on a person, there's a barrier. But we thought we could overcome that barrier, and here's how. So the first was, how can we have a low-cost source of electric pulses? And that got us thinking about lighters. So why do we think about lighters? Well, the way a lighter works is that there's a gas supply, but you need a spark. What you make a spark is there's a little piezoelectric element inside. Piezoelectric elements interconvert between electricity and mechanical motion. So the mechanical motion of a hammer hitting the piezoelectric crystal generates an electric pulse. That pulse in the lighter goes across an air gap and makes a spark. And you light your flame. We don't want the spark, but we do want the electric pulse. So we go, look inside one of these things. First, you can buy them, get on Alibaba, and for 30, 40 cents, you can buy one as many as you want. So they're made by the billions per year. And in fact, if you look inside, we don't need that whole thing. We don't need the gas and all that. We just need this little piezoelectric crystal inside, which is essentially free. So we go from $10,000 to an essentially free voltage supply. Doesn't require any batteries. You just, your thumb, and that's all it takes. So that led to the design of this electroporator. Everything from here on up actually is just buy that. And it's not the one for a lighter like I showed. It's actually for, maybe you've seen these, I know I think it never gets cold here, but if you go to a place where it does get cold and you sit outside, they have these gas heaters that go over to keep you warm when you're at your cafe in Paris or something. And it has a little bit bigger, more robust lighter. So we just buy that. And then at the end, we put the microneedle electrodes, which I'll comment on in a moment. So using that very simple system, we use a piezoelectric voltage source. So plug it in. The whole cost to make one of these things should be less than $1. It's handheld, thumb operated. It's like the size of a pipetter. There's no battery. There's no power supply involved. It's targeted to the epidermis, and this is where the microneedles come in. So the microneedles are short, so they don't penetrate deeply into the skin, and so therefore your electrodes are just below the surface. We avoid the nerve stimulation. We can electroporate the cells in the epidermis, and that's where most of the cells are in your skin, so that's where we want to be. And we avoid those nerves that are found deeper down. So these microneedle electrodes allow us epidermal targeting, avoid the nerve stimulation, and we have used these onto, us researchers, but also on naive subjects who are blinded. We then either gave them the pulses, we get 10 of them, or we do the same thing except the wire is disconnected. So it just goes click, 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 and there's actually no voltage, and the subjects can't tell the difference. If you look at the skin, you see just the mildest little twitch that happens, or you might not even see anything. So we've been effective in localizing the electric field to avoid that nerve stimulation. So we think we brought the cost way down, simplicity is good, and we think we've avoided this nerve stimulation issue. You don't need someone holding the subject down. And so there's no lipids, there's no viruses involved. So here's what we can see. This is in the rat where we delivered DNA that encodes for green fluorescent protein. We're looking at a view above on the skin as well as then a cross section through the skin. And we can either use clamp electrodes with a conventional electroporator, the $10,000 box, or the microneedle electrodes with the piezoelectric pulsar. And you can see in both cases we're getting transfection to occur, we're delivering the DNA, and it is really nicely localized into the epidermis. We then carried out a vaccination. This was a few years ago, so of course it was a COVID vaccine that we used, and this was again a DNA vaccine. It's a prime and a boost protocol, and we then can look at the antigen-specific IgG responses in the biopsy mouse. So we either gave an intramuscular injection of the DNA vaccine at 10 or 100 micrograms, and you can see the 100 micrograms gave a stronger response than 10 micrograms, not surprising. We give an intradermal injection of 10 micrograms, about the same as intramuscular, 10 micrograms. And then finally, change the terminology, so it's also sometimes called the electropen. In any case, the electroporation approach that we developed with 10 micrograms with electroporation, we're now getting a response like 100 micrograms without the electroporation. So this was some proof of principle that we could use the device and enhance the delivery. So we get to topic number 4. And now, as I mentioned, we can also use microneedles in reverse. Oh, so I guess I should be saying, since this is the Venture Center, I should be saying a little more about the translational activities. And so in what we just discussed, so this led to the formation of a company called Piezo Therapeutics. And that company is early stage, 3 employees, does have a first seed round of funding to get it going. Actually, it's been very efficient with the money. It's generated really nice data on DNA and now on mRNA vaccination as well, mostly as a partnership model. So developing the technology for delivery and then partnering with companies that have DNA and RNA that they want to deliver and co-developing the product.
Is this the one with SAR?
This is with sod, yeah, sod bomb. Who's that? All right, so the next one is the microneedles in reverse, where we make some pathways in the skin and pull things out. So what we're thinking about is that blood is a really great source of biomarkers, and it's used all the time in medicine. But blood is just 8% of the fluid in the body. There's a lot of other fluid there, and it might have some other advantages and some other information that we don't get in blood. Blood is super convenient because it's designed to flow. You just poke a hole and out it comes. Very easy. Intracellular fluid, that's a little more complicated. You're going to probably kill the cell if you're going to try to get any fluid out. But there is about a quarter of the fluid in the body is the interstitial fluid. So that's the fluid that's in the extracellular spaces. And that fluid is related to the blood because that's its source. Blood flows into the capillaries, fluid exudes out of those capillaries into the tissue space. The tissues then metabolize, they remove things, they add things, change it around. That fluid then flows through, mostly into the lymphatic system and then back into the bloodstream again. So the interstitial fluid should be related to blood. It should have information that you can also find in blood because that's its source, but should also have some other information due to these metabolic processes happening in the tissue. So that's our motivation. To do that, we're going to use microneedles, and the reason is that if you poke a hole in the skin and try to get some fluid to come out, you'll probably get blood to come out, as you know, right? So we need to make the poking that we do has to be very gentle. So we make these needles, they're just 200 microns long, and they make a very gentle and small puncture into the skin, after which we apply a mild suction and pull the fluid out. So we have to play this careful game of perturbing the skin enough to get the fluid to come out, but not so much that it causes bleeding. And here you can perhaps see some interstitial fluid that has been drawn out of the skin through some of these micropores. The fluid is, of course, clear, so it's a little bit hard for you to see it, but maybe you can make out the shiny droplets that are on the skin surface. We can get out microliter quantities of fluid. So single-digit microliter is sort of the standard. If we play some special tricks, we've been able to get into tens of microliters of fluid, but we're not getting milliliters, that's for sure. So we did a study in a small number of healthy subjects, 21 subjects, again, from the Atlanta area, and we collected 3 different fluids. We collected the interstitial fluid, as I described. We also collected suction blister fluid. So as the name suggests, we apply heat and suction to the skin. It makes a blister, pop the blister, collect the fluid. That's something like interstitial fluid, so we thought that was a useful control. And then we collected blood by venipuncture and measured what was in the plasma. We then used mass spectrometry to identify compounds, and we found more than 10,000 different compounds among these samples. Among those, there were 69% that were common to the interstitial fluid and the plasma. And as I mentioned, that's maybe not surprising because the interstitial fluid comes from the plasma originally. We then found that 11% of these biomarkers were in the plasma and not found in the interstitial fluid. And that, I think, is primarily due to binding. I should know, these are all small molecule metabolites that we're following here. And so if you have also a very high molecular weight compound, that's going to have a hard time coming out of the capillaries as well, but that's not an issue here. So I think these might be, albumin bound or bound to something so that they don't come out of the blood easily. But I think most interestingly is that 17% of these were found uniquely in the interstitial fluid and not found in the plasma. And this reflects these metabolic processes happening in the tissue. And so this means that if you're looking for a molecule that is in the blood, but you want to also find it in interstitial fluid, you probably will, because the large majority of plasma molecules are also in interstitial fluid, in a few cases not. And if there's some molecule that you can't find in the blood, you may be able to find it in the interstitial fluid, and that could open up some new options. So I think the main opportunities here are twofold. Interstitial fluid doesn't clot. Blood does. And in fact, the only thing that is routinely measured in the interstitial fluid is glucose in the continuous glucose monitors. In that case, a biosensor is inserted into the subcutaneous space, sits there, and continuously measures glucose in the interstitial fluid of the subcutaneous space. You couldn't do that in blood. If you stuck something in the bloodstream, it would cause a clot, it'd be faster. So you could, but you can put it outside the bloodstream, and so that's an advantage. we don't want to stick sensors in the body. That's much more complicated. We'd like to pull fluid out, and then you do whatever you want with it. You can measure it in the patch itself. You could take it into a point of care device. You could send it to a lab. You're free to do whatever you want at that point. So one opportunity is if you want to make continuous or frequent measurements where avoiding clotting would be advantageous, interstitial fluid may be a good choice. And the other is if the biomarker you want is one of these that you just don't find in the blood, well, then clearly it's a good choice. And the obvious place to think there is in dermatology. You could do some diagnostics in dermatology that might otherwise require, say, biopsy. You could avoid having to do a biopsy. So we did do an experiment where we wanted to follow pharmacokinetics in the interstitial fluid and the blood just to see how they compared. And we used the favorite drug of Atlanta. Atlanta is the home of Coca-Cola, so caffeine was our drug of choice. And we gave people, gave them Diet Coke, either with or without caffeine. They drank it, we followed them, their response. And what you can see on the black data points is those who drank the caffeinated beverage, either in the interstitial fluid or the plasma, that there was no significant difference between the interstitial fluid and the plasma. And then the white points are the background for those who drank the non-caffeinated Coke. We can then calculate the various pharmacokinetic parameters, and may or may not be interested in them quantitatively. The point I want to make is when you do the statistical test, there's no significant difference in these parameters for interstitial fluid and plasma. And that's because caffeine is a small water-soluble molecule, and it readily escapes capillaries and gets into the interstitial fluid. So we come to the final story.
So
Yes.
just on the previous slide, when you were talking about pulling the interstitial fluid, right? Is there a possibility of contamination coming with blood coming in, or how did you design your needle in such a way that you're actually destroying the interstitial fluid?
So the question about contamination of interstitial fluid. So there really are two sources of contamination. One of them is blood. And so we avoid that by the gentle method that we use. So we do puncture the needles in, and we don't completely escape capillaries. So some capillaries are burst in the process. What we may do is wait a minute or two and then apply the suction, or we may slowly ramp up the suction. And so that gives some time for a little clotting to occur and those capillaries to be at least sufficiently repaired that the blood doesn't come out. So it's a little, little optimization that goes on there to make sure that the blood doesn't come out. The other is that there could be some contamination on the skin surface. So you do need to make sure you clean the skin well first, but that is a possible source.
Mark, there was an online question on this also, which is how do you make sure the fluid collected is not a mixture of interstitial fluid and sweat glands? Sweat will dilute ISF.
Yeah, so sweat would be another source since it's secreted onto the surface as well. So you certainly should clean the skin first. So any prior sweating is not going to be an issue. But the question is, what about sweating that happens during the collection period? So there, quite frankly, there's nothing we can do about that. So the collection should be done as quickly as possible and in an environment hopefully where people aren't sweating. But admittedly, that's not our current method of collection does not address that problem. There's another way of doing it though. So right now we just make holes and pull fluid up onto the surface. And others you can make microneedles out of a hydrogel material. So that's dry, you stick it in the skin, and then the hydrogel pulls in the water and it swells, and then you take the needles out. So then you're doing a collection truly within the skin. There should be, the needles don't contact the surface of the skin. So then you don't avoid the blood necessarily, but you definitely do avoid anything that's on the surface to contaminate.
But the AUC values are very different. Like the rest of your parameters are almost comparable. AUC, how did you compute them?
Yeah, the error bar is large enough that that by a t-test they were not significantly in our small sample size.
Please continue.
Okay, so last topic is the hollow microneedle, and now we're leaving the skin. I've advocated that these microneedle patches, you administer themselves, they don't hurt, do it at home. That goes away at this point. This is not for self-administration. This is not painless. This is an injection into the eyeball. So it's a different scenario. This is done in the doctor's office under, you get a topical anesthetic before you do it. It's a different situation. And the reason that we do this is also different. It's really about targeting. It's about targeting the drug to a particular part of the eye that I'll explain, and the microneedle enables you to achieve that target. So here's a scenario that motivated this work, and that relates to steroid delivery to treat macular edema, so that is swelling in the back part of the retina. And we would benefit from targeting. So what's done today is here's a cross-section through the eye. The cornea is shown here on the top, the lens is kind of in the middle, and the optic nerve is in the back, and there's a yellow region that goes around the periphery, that's the retina. So if you want to deliver a steroid to the back of the eye today, a hypodermic needle will be inserted into the eye, and a drug will be injected, for example, a steroid like triamcinolone acetonide, and that drug, some of it, will go to the back part of the eye, which is its site of action, and it will deal with the inflammation. But it'll also get into the front part of the eye, and it can cause cataract, and it can cause glaucoma. So we'd really like to have better targeting. For example, if we could get the drug just in the periphery in the back of the eye, where you need it, then you could avoid these side effects that happen when the drug gets up into the front part of the eye. So how do we do it? Well, you know the answer. It's a microneedle, and a little more on the ocular anatomy explains it. So the very outer layer of the eye, the white part of the eye, that's the sclera. That's a structural tissue. Interior to that, the next layer is the choroid. That is a vascular tissue. And then comes the next layer, the retina, and then the interior is filled with this vitreous humor, which is just an inert hydrogel environment. So if you want to treat the back of the eye, what you're really talking about is treating the choroid and or retina, depending on what it is you're treating. So if you take a microneedle and you stick it across the sclera, which is under 1 millimeter thick, and make an injection at the interface of the sclera and the choroid, the path of least resistance for fluid flow is in the gap between those two tissues. Choroid is a very tough tissue. Sclera is a very tough tissue. Choroid is pretty tough as well. But the two tissues are not tightly attached. So when you make an injection at the interface, it wants to flow in the potential space between those two tissues. And so you can achieve the kind of targeting that we're after. Here's a picture of one of these microneedles shown next to a conventional hypodermic needle used for intravitreal injection. And if you take a closer look, you can see, so this needle is about 0.5 millimeter in length. It's made of stainless steel, looks a lot like a hypodermic needle, but it's really small. And in fact, quite frankly, the way that we make it is, we buy a 30-gauge hypodermic needle and we cut it off short. So it's actually pretty simple to make. To make it sufficient quality for human use, you do a little more effort, but that's the basic way to make it. And when you take one of these and inject it into the rabbit eye, we then can take a cross section. The anterior segment is here. The cornea is shown on this side. On the back would be the optic nerve. You can see the site of injection with a little bit of leakage in this case, and then this thin band of fluorescence where we've injected into that gap between in the suprachoroidal space. So jumping way forward, the company that we started, Clearside Biomedical, has proceeded to work on this, gotten through clinical trials, and had a product launch in 2022, a product called Zypier, and it's triamcinolone acetonide. So this steroid, it's a very water insoluble steroid, so it takes about a month for it to dissolve. So you don't want to give suprachoroidal injections every day, so you want a slow release formulation. So this triamcinolone acetonide is injected into the suprachoroidal space and is able to provide an extended treatment of swelling in the back of the eye. And that brings me to the end. So this idea of making things small and being able to use that for various advantages in medicine, whether it's the dissolving microneedle patch to facilitate vaccination, the star particles for treating larger areas for topical, say, dermatological delivery, using microneedles as electrodes to give electroporation pulses, using microneedles in reverse to pull fluid out, or finally getting targeting in the eye with a suprachoroidal injection. So let me acknowledge other of the lab members who have done the hard work that I am presenting, also to acknowledge the funding sources that we are able to, that enable this work to take place. And if there's time for questions, I'd be happy to take them.
Well, first of all, thank you very much. What I'll do is I'll take a few questions online first.
Okay.
Because I think others had an opportunity in between as well. So there's a question. I'll start first with a question from Sagar. Have you used it for animal diseases yet? And if so, what are the kind of design issues, for example, depth, the needles, animal microneedles, and so on?
We have, of course, pressed microneedles into animals much more than we press them into people. Those animals tend to be, though, bright mice and rats, so not as interesting. For veterinary applications, we did do a study where we teamed up with an animal health company, Marielle, now Boehringer Ingelheim, that they were interested in rabies vaccination. So we did a study in beagle dogs. We applied the patches to the inner ear, because fur becomes a problem on animals that we don't have so much in humans. But the inner ear tends not to have fur on it. It's pretty easily accessible as well. I was told if we wanted to work with cows, the site recommended to me is under the tail. That's a site without fur. I'd rather do the ear. That seems easier to deal with. Anyway, so we did this study in the ear, and then the bottom line is, yes, we were able to do the vaccination, and it worked well. A project that we're doing right now is for veterinary scenario, but it's not to deliver a drug, it's to deliver tattoos. We're working with an organization that is developing non-surgical sterilization methods for dogs and cats, and they're particularly interested in free-roaming animals, that wander around town and no one particularly takes care of. So they need to be sterilized, and you need to know when an animal has been sterilized or not. So you could clip the ear or put on a tag on the ear, which is so nice for the animal. So we're again using the inner ear, but putting tattoo ink in the microneedle patch. So you press the patch on, and then you get a little square or circle or whatever shape you want to make, so that the animal has now been marked and know that it's been sterilized. So I think there are opportunities in veterinary health as well. The question for any innovation, quite frankly, is there a market for it? Will the market pay the costs? And in human health, that's easier. In animal health, well, if, dogs and cats, sometimes there's money there. If it's, in a non-vegetarian scenario, if there are animals that are livestock, cows and pigs and whatnot, that's a lot harder. There's not a lot of margin in those animals. And so I think that, and it goes even one step further. You want to do it on plants and agriculture. So it gets harder and harder as the opportunity to spend money on something becomes less. So human health is the easier scenario to think about it.
I think the feedback was from my thing, but that reminds me, our team has put up a feedback link on the chat box, so those online can fill that, and those who are sitting here will need to use that QR code at the end. Okay, so Kedar has a question online, which has got to do with a comparison between microneedles versus other technologies like emerging jet injectors and so on and so forth. His question is how do microneedles compare with emerging jet injectors that are also positioned as painless delivery options, pros and cons of both the options. We see in literature speaking about intradermal delivery of insulin being equally efficacious, but do you see commercial products in this route also, and why? Yeah.
I think that there are various ways of getting molecules across the skin. So one of them is the really conventional ways with chemical enhancers. So you mix some chemicals into your formulation, and that helps the drug get it better. And that's what's in transdermal patches, and that's what's in conventional, non-microneedle conventional patches. That's what's in conventional creams that you rub on the skin. And it works, but it only works to help a little bit. That is a drug that gets into your skin some, gets into the skin more. But you can add your chemicals to insulin. Forget it, it's not going to go in. The vaccine, it's not going to go in. So you need something more aggressive. So you can get more aggressive. You can use ultrasound, you can use electric fields, and you can use jet injectors. There's all kinds of things. And these are devices. These are big things. They have batteries associated with them, they have complexity associated with them, and a cost associated with them. So that's a barrier because they are really devices of size. Now, microneedle patch may be classified as a device by regulatory authorities, but they're basically like a patch. And so you don't have that kind of complexity and cost. You have something that's just really easy to use, and you don't plug it in, and it's not going to fail because it gets wet or all these things. There's nothing reusable that you have to use because it's too expensive to make it single use. So I think it sits nicely In between, it's a physical delivery method, and so it's effective to deliver the drug. But it's also, it looks a lot like just a regular pharmaceutical. It looks like a patch, costs like a patch, feels like a patch. And so I think that's why it sits in that nice space. I should note that conventional jet injectors, the general, what's been reported about them is that basically their main advantage is that they're not needles. And for people who don't like needles, they're afraid of needles, they're not needles. But in every other way, they're not better than needles. Still a liquid formulation that you have to use. The injection hurts. The injection causes bleeding. If the injection isn't done at the right angle, you get a wet injection. Some goes in, some stays on your skin surface. It can be loud, the pop of what's shooting into your skin. And so jet injectors have been around a long time, decades. They're hardly ever used. And the reason really is that they're not better than injections, other than the fact that there's no needle involved. That may be it. I think that's an objective point of view, but some may disagree.
Did you want to comment on intradermal delivery of insulin?
Intradermal delivery of insulin. Yeah, so there has been some work on that. We did some, Beck and Dickinson did a bunch more in human subjects, and what they found, interestingly, is that if you administer insulin into the skin, there's a very rich capillary bed in the superficial part of the dermis. And so you can get quicker absorption. So if you look at the pharmacokinetics and the pharmacodynamics of insulin delivery in diabetics, and you compare it to subcutaneous injection conventionally to intradermal injection, there's a faster onset when you give intradermally because you're accessing these capillaries and the insulin gets in the body faster. And so that can be really valuable, especially in a continuous insulin delivery, in a closed loop, say, feedback insulin delivery scenario where you get information about blood sugar and then you want to infuse some amount of insulin in response, and you have this circular loop of get glucose information and deliver the insulin. As soon as you get the glucose information, you want insulin in the bloodstream immediately. But in a subcutaneous injection, it's about a 45-minute delay. And that's why control of glucose levels, not by the pancreas, but by things on your body surface, they are imperfect because they're always behind. To get information, the insulin's not in the bloodstream until later. So if you can shorten that time, then there's some real advantage. You close the loop even tighter, and it may be that intradermal delivery can enable that.
I think I'll leave out a couple of questions, but there are a couple of questions on applications. Are there any applications in wound healing? And the second one has to do with melanoma, but I think you've shown one example as well. So I presume we can leave that aside, but are there applications in wound healing is one of the questions.
So wound healing, a distinction of the skin wound is that probably the skin barrier has been breached. There's an injury there, and often it's open and wet and so forth. So the goal of getting across the stratum corneum has now disappeared, because the stratum corneum is already gone. But nonetheless, there may still be reasons to use microneedles, and one of them is a mechanical function. So you can use it kind of like staples. So it can help to seal a wound, and the microneedles go in, and you don't want dissolvable ones now, you want ones that anchor, and so you can mechanically use it as a wound closure device. And then you might, in addition, incorporate a drug into the microneedle, maybe even for a slow release. You didn't really talk about that unless when we were talking this morning, but you can also formulate these microneedles to slowly release drug over time. And so you can have a local treatment with antibiotic or growth factors or whatever it is you might want to deliver. And in that way, the microneedle could be easier as a depot delivery system. For melanoma, I think we have to be very conservative. So certainly the dermatologists are. For non-melanoma skin cancer, you certainly do want to get rid of it, but it generally does not go systemic. Whereas melanoma, that's the big fear, is that it can go systemic and then becomes much more dangerous. So at least right now, the standard of care is melanoma, you cut it out. No discussion, you cut it out. And so we're not really targeting melanoma. We did use the animal model because that was a convenient model to use. But I think melanoma may not be an ideal target, say, for star particle or microneedle patch delivery, because surgery is really what's indicated. But non-melanoma skin cancer can be treated with like 5-fluorouracil, and there I see a bigger opportunity.
Sorry, I'll just continue with a couple more questions. There's one about limitations. So let me just club two questions. There's one from Supriya, which has to do with are there limitations of what kind of drugs can be delivered, what kind of vaccines can be delivered? Presently approved vaccines, like you have been doing it with Serum Institute, right? Are there some limitations in terms of what you can work with and what you cannot, in terms of perhaps the solubilities and also that you mentioned? But the other part of the question is, are there limitations in scaling up and doing a lot of millions of such needles, right, in terms of microneedle technology?
The primary limitation, if you want to deliver some kind of a drug with a microneedle patch, is dose. The microneedles are small, and that's good, but it's also bad. And it's bad in that the total dose you can deliver, if you're under 1 milligram, that's the sweet spot for it. You might get into the 1 to 10 milligrams range. That's really the cutting edge of the field, is to get in that area. If your drug's 100 milligrams a gram more, the microneedle is not the way to do it. So that's going to be always the first cut. For vaccines, never an issue. Vaccine doses are always extremely low. So pretty much all vaccines are on the table from that perspective. The next question would be, can you formulate this drug into a microneedle? So it involves drying it and keeping it active in the drying process and afterwards. Small molecule drugs, yeah, you're probably okay. Proteins, vaccines, it's a little more complicated to dry those. We've worked with a few dozen different vaccines and a smaller number of protein therapeutics. We haven't worked with one yet that we haven't been able to incorporate into a microneedle patch. Some are easier and some are harder. Some are a lot harder. Like the measles and rubella was quite hard because those are live viruses, and it's 2 different viruses, and got to make them both happy when you incorporate them in. So there can be a lot of work to make it work out, but I think if you can satisfy the dose requirement, probably if you work at it a bit, you can get the drug in.
So the final question online before I invite a few other questions. Question on star needles for atopic dermatitis skin is already very irritated. For effective delivery, will need a pressure rub, will make it worse before it can be effective. Did you try suprachoroidal delivery to deliver anti-VEGFs in wet AMD?
Okay, so those are two different questions.
Let's go to the first question, which has got to do with the pressure rub that's needed, and will it make it worse if you're using star needles? And the second question was about the suprachoroidal delivery.
Yeah, so if you're using the star particles, you do need to rub the skin.
Yeah.
And in some skin diseases, the skin is very fragile, and rubbing vigorously would probably not be a good idea. So I think for those really the most fragile ones, ones, for example, where the epidermis is no longer well attached to the dermis, there's some scenarios there, and if you rub it, you may strip off the epidermis. I certainly wouldn't start there with star particles. So although I don't think with atopic dermatitis, I mean, there could be some irritation that results. The main application that we're going after, I didn't comment on this, there's a company that is commercializing this work, and Alice Pharmaceuticals, and their main emphasis is on siRNA delivery in combination with star particles. And the siRNA is designed in such a way that it's really long-lasting, lasts for weeks. And so you have a single rubbing on the skin, the siRNA gets into the cells in the skin, knocks down the expression of some protein that's problematic, associated with inflammation or something in the skin. And so the idea there is that the treatment may be once a month. And so in the ideal situation, it's not at all irritating, but maybe it is a little bit irritating to the skin. If you only do it once a month, then that may be tolerable. There's also about anti-VEGF.
Yeah, anti-VEGF and also suprachoroidal.
With the suprachoroidal delivery. So a key principle in the suprachoroidal delivery, a key limitation is you can't do it every day. You have to do it quite infrequently because you have to go to the doctor. VEGF, anti-VEGF drugs are given every month or every couple of months right now, but that's because they're injected into the vitreous. So I said the vitreous is a bad place to deliver drugs. You want to go to the suprachoroidal space. Sometimes it's a good place to deliver the drug. It's avascular. So you inject the anti-VEGF drug into the suprachoroidal space, and it sits there, and it slowly diffuses out to the retina, and it can get to the blood vessels that are there and ultimately get cleared. But you've created this depot in the vitreous that lasts a long time. If you give conventional anti-VEGF drugs, which are antibodies, you give it into the suprachoroidal space, there's a lot of vasculature right there, and it'll be cleared out in a day or within a few days. So conventionally formulated, say, proteins or antibodies are not, I think, a good idea to put into suprachoroidal space. You would have to formulate them in such a way that they would be slowly released. Now, Clearside mentioned the product that they developed. Number 2 that they're working on is an anti-VEGF treatment, but it's not using a conventional drug. It's using a small molecule drug, and also one that is very water insoluble. So a single injection of these particles can last for a month or longer, and so that is where there's a good match. Suprachoroidal plus slow release is the combination I think we need.
Great. So I think many of you will have the benefit of talking after the event, but I thought if anybody has any burning questions, yeah, we have some there. So a couple of them we'll take.
First of all, thank you for the wonderful presentation and the slides. Very, very simplified way of presenting a difficult topic. My question is, what about can these microneedles be used for CNS related like diseases, whether they can cross the blood-brain barrier? They could try it.
A key feature of the microneedle is that if you're presented with some surface, you can get just across whatever barrier there may be on the surface and below it. So stratum corneum presents such a barrier. And in the eye, the sclera presents such a barrier. So when you get to the CNS, that's deeper in the body. So you're not going to, I don't think, press a microneedle in, have it go through your skin, through your skull, and hit the brain, right? They're not going to, well, they're short, and they're not designed to go that deep, plus in the skull, it'll never get through that. So if there's a scenario where in the context of surgery, the brain is exposed, and then you would like to be able to administer something to the exposed brain, then a microneedle patch makes sense. But that's I think, a very particular situation. It's not the more general situation like you could have the skin or the eye or some other surface. Now, maybe what you're thinking is you could go intravascular. What if you put some particles into the bloodstream and had needles sticking out and they could poke into the blood-brain barrier and help drugs get across? Conceptually, yes. I think, though, actually making those and making them work, it gets quite complicated.
Quite risky. Your last.
Hello. Can any of these microtechnologies that you mentioned be used for studying something fundamental like cell migration, either to prevent bad things like metastasis from happening or to study homeostasis? So which one of these would be the closest to study cell migration?
Yeah, I'm not sure I know enough about the issues in cell migration per se, but I can comment sort of more generally how you might work in some sort of in vitro setting and help with some basic research in, say, in cell biology kinds of scenarios. So one way is that the microneedles have been used for intracellular delivery. So you make them much smaller, and then you can have the microneedles contact cells. So you can either have a monolayer of cells and bring these, they're called nanoneedles often in the literature, even though their dimensions are still small microns. They bring the nanoneedles down and they can puncture the cells, or you have the cell, the microneedles sticking up, and you bring the cells to the microneedles and puncture them. So that's a way to put things into the cells. You can also use these microneedles as electrodes. So now you have a microneedle that's penetrated into the interior of the cell, and so now you've got an electrode inside the cell. You can have another electrode outside the cell, and you do electrophysiology kinds of measurements with that. That has been done probably most extensively in brain tissue or other kinds of tissues with electrical activity, where you can now have each microneedle as its own electrode, and you can map out electric fields, either stimulate, but more typically collect electrical measurements. And with very nice spatial resolution, you can see how the electric field is varying in space and time with your array of microneedle electrodes. So there are things like that can be done.
I think one last.
I want to know about the concentration of the drug. If this works very efficiently, then can the drug concentration be reduced, or you need more, particularly for the start particles like bifluorouracil, etc. So if we can reduce, is it possible to reduce the concentration? That
So-
relates to the efficiency of this.
For the star particles, for whatever amount of drug you put on the skin, more of it will get into the skin because we made holes so that the drug's able to get through better. So your goal may be to get more drug because right now you put the drug on the skin and just not enough is delivered and you need more to be effective. But you might have an alternate goal, which is you can actually deliver enough drug, but you'd like to have less, you save money, for example, so you could put a lesser amount of drug on the skin, but use the star particles, still get the same amount into the skin. So depending on what your goals are, you could do that. In the case of vaccines, you can have dose sparing. So as mentioned, the skin is more immunologically active. So if you have a vaccine that is effective in the muscle, you could give less of it in the skin. And in fact, it's been shown with rabies vaccine, and India may be the only country in the world where it's actually formally approved to use rabies vaccine with dose sparing. So if somebody needs rabies vaccination, they get a dose intramuscularly, or they get, it's either a fifth or a tenth. I think it's a tenth of the dose intradermally. And so that would be a dose sparing scenario because the skin's immunogenicity for a different kind of reason.
Well, I wanted to conclude, but there's a question from students. Last one. We have lots of questions, but I have to stop. Go
I was
ahead.
just wondering, could this microneedle therapy be used for, say, cancer cell ablation if we incorporate some sort of nanoparticles into the microneedle patch? And then we kind of, say, we utilize photothermal therapy or any sort of therapy to immobilize the nanoparticles into the cancer site. So could we use that to kill cancer cells essentially?
So the microneedle can deposit material into a tissue. If that material is a nanoparticle, that's fine. The microneedles are micron scale, so nanoparticles can be incorporated, have been incorporated into microneedles for delivery. If you wanted to deliver those particles into the tumor and disperse them, say, throughout the tumor, then a microneedle might be advantageous. You've got lots of needles that puncture all in. If you just give an injection, you may not get perfusion throughout the whole tumor. The microneedles could go in, and you might make them bigger so that they go all the way deeply into the tumor and then deposit all over the place within. But you do need access to the tumor, right? If it's skin cancer, you're in good shape. But if it's in the liver, you're not going to get the microneedles there so easily.
Right, so something like skin or breast cancer, something that's superficial, only then.
Yes, basically, if you can access it, if you can touch it, then the microneedle is going to work well. But if it's deep, then, well, you need to do something to be able to get it in. It could be laparoscopic, for example. So there are possibilities.
Okay. On that note, let's close, right, formally, and we'll break for tea. But before that, those who have to fill up the feedback form online, please do. The others should use this QR code. And we close by formally thanking Professor Mark Prausnitz again